I would like to talk about KC&Q channels, and virtually they are already forever. So I'm working with these channels since I was a PhD student, and that's quite a time ago. So these are really old dogs, but we always keep on finding new stuff. And this is about KC&Q1, so the first one cloned, and I'm working on this since 1997. Okay. So these channels are expressed in all different kind of tissues. Just here I mentioned some of them, which include the heart, the inner ear, the thyroid gland, and others. And the channel serves their all different kind of functions. So which is a very broadly expressed and very functionally important channel in different settings. Today I will concentrate a little bit about something which might be particularly important in the heart. And when this channel contains loss of function mutations, they can cause a long QT syndrome. So what is long QT syndrome? Just to make it very briefly. So these are loss of function mutation causing a decrease in channel function here. This decrease in channel function leads to a delayed repolarization of the ventricular cardiac action potential. So the current is lower during an action potential. This ventricular change, so the repolarization change becomes obvious in the T wave of the ECG. And sometimes in these patients, often triggered by beta-adrenergic stress, you can see something like missing waves to early waves, which then can trigger torsade-dependent arrhythmias, which really undulate around the isoelectric line. And then, of course, a heart which beats like this fast is no more pumping correctly, the blood pressure goes down, no perfusion of vital organs, and then you can die. So that's quite a serious problem when you have that. So it was reported already in 2005, for example, by Park et al., that binding of PIP2, there we have the molecule again, we have heard about that yesterday already, that reduced binding of PIP2 is associated with loss of function in this channel, which cause some of the phenotypes associated with long QT syndrome 1. It's just one part of the mosaic there. So how does that work? So PIP2 interacts, and particularly much with the S2 S3 linker, a little bit with a lower S6. There is some involvement also from the S4 S5 linker. So here is where the PIP2 is binding. When we talk about PIP2, normally we mean this high abundant PIP2 which is present in the plasma membrane, which is PI45P2. So however, we also now have even a structure for that. But there's another spice of PIP2 which has a different phosphorylation pattern. So the PIP2, the phosphate group are associated with position 3 and 5. And when I was younger, I did this study and found or identified in the S0 helix, which just precedes S1, three disease-associated mutations which are not stimulated by this kind of PIP2. This PIP2 is generated in intracellular vesicles. It's not present or hardly present at the plasma membrane, but it's exclusively generated by one kinase, which is called PIC5, in intracellular vesicles. And production of this PIP2 triggers exocytosis, which contain these channels. So what do we know now about the PIP2 binding? So this is the PI45, the more common plasma membrane, PIP2. How does it work on the channel? So last month there was a nice paper from Rod McKinnon, and they used vesicles incorporating the channel, and under resting conditions, so PIP2 cannot interact with its interaction site, which is here at the lower voltage sensor and close to the S4 S5 linker. So then they polarized these cells, pushing the voltage sensor in the up position. Now there's conformational changes here in the PIP2 side. We now generate by this conformational change an affinity for PIP2, and when PIP2 binds, PIP2 basically triggers the port to open. So it's basically like binding of this ligands open the channel. It's not directly that the voltage sensor does the job by itself very easily. So, but what about the other PIP2? You know, this trafficking modulator PIP2. So I'm working since several years on that, and what we figured out is that there's a kinase cascade, serum and glucocorticoid kinase, phosphorylates and activates this PIK5 kinase, which in turn generates this PI35P2 in intracellular vesicles, which then triggers exocytosis of endosomes containing the channels. Via this mechanism, the density of the channels at the plasma membrane is increased, and that's how we generate more current. So it's a different mechanism. That's very clear, and it's under control of a small GTPase called RAB11. So then we did this recently, these experiments. So when you inject water-soluble PIP2 analog into oocytes expressing this channel, you can see that the current is increased. So from here up to there. You also can co-express with a PIK5 kinase, which is upstairs and generates PIP2. It generates a similar increase in current. When you do both manipulations, you get an increase, but no additive, no synergistic effect, indicating that these are really in a cascade behind each other, so which made it easier for us to use it as an assay. Overexpression of that, just this kinase generates this PIP2. We can increase PIP2 and use it as a much easier assay. So we did this, and this is what you get. And when you then knock out key residues for the PIP1 site, where the other PIP2, the 4,5, binds to, you can see you can also knock out completely the effect of this kinase, of this PIP2. However, we also had charge replacement or neutralization here in the S0 helix, and also there we see some modulation. So this arginine obviously is not directly involved in some interactions. However, there's also one arginine, when you mutate it, then it can inverse the effect of the PIP2. So something really seems to go on with S0. Okay. So then we wanted to understand, is there physical interaction of this S0 with this PIP2? So what we did is we generated a synthetic peptide resampling this S0, the CD spectrum, and also NMR, 2D NMR suggested it's really folding a helix. And then we also did here a 1D NMR, and you can see there are shifts when we apply this water-soluble PIP2. So there's physical interactions there. So we also wanted to get an idea about specificity of is it only taking this PIP2 or also other lipids, membrane lipids? We conducted this fluorescent-based PIP strip assay. So there are different lipids immobilized on some paper tissue, and we just used fits attached to this peptide and put it on, washed the not bound PIP2 off, and you can see there is some lighter parts in the middle. So PIP2 is the peptide is binding there. However, in the blank, this lighter part is not there. We repeated this 5 times, and you can see it takes almost all the lipids. It's not really specific. However, there seems to be a slight tendency maybe for the 3,5 PIP2, but it's definitely not specific. In this assay, note this is not a Western-based assay, so the noise to signal ratio is much weaker with this fluorescence assay. So we also checked S4, S2, S3, and you can see there seems to be some binding here in the PIP2, PIP3 region there. We also did it with S4, nothing there. S6, maybe a tiny little bit. We used an S0 equivalent from S4, which is not sensitive to PIK5, and there you can see nothing. So basically there seems to be really physical interaction. We wanted to explore that further. We scanned the whole S0 and checked for the stimulatory effect of the PIK5 kinase. And you can see there's an area in the N-terminus, which is really at the tip of F0, which is here, which really seems to be important. And then there's some effects also in the middle. Okay, so there's interaction. And what would PIP2 do? PIP2 might be or function like an anchor in the membrane, positioning S0. In order to get an idea what positioning of S0 relative to the inner membrane plane in a leaflet really is effective in regulating the channel, we applied another assay. So what we did now is we used our cysteine mutants, injected cadmium, and cadmium can be coordinated with some affinity between the negatively charged lipids heads and the cysteine. Via this, we generate a force pulling the helix to the membrane border. And when we did this, you can see pulling the helix on one side closer to the membrane is inhibitory. On the other side, it's really stimulating the channel. So it's really positioning the helix to the membrane really modifies the channel. So then we went into MD simulations, and this is basically what we got. So we believe that there are two PIP2 binding sites, the one which is reported, and then an additional in the outer S0. So, but what is that good for? So when both PIP2s are present in these models, one can see that virtually the affinity at the second binding site, so this one here, the known already, is increased. So here the numbers, lower numbers mean higher affinity in silico, in the computer. Okay, and this is the mechanism which we have identified in silico. So when PIP2 is added, this PIP2 at S0, the new binding site, really repositions S0 relative to the lower part of the voltage sensor, which in turn positions three residues, at position 111 of S0 relative to arginine 116, which is a direct interactor with the other PIP2 as well, and another arginine here at position 189. So, and really stabilizing this triangle here, which boosts and promotes the affinity at this binding site. So here you can see that the distance, so the positioning is really changed between this residue here to there, which is green. So here in green, when two PIP2s are there, the distance is reduced. The other distance is also reduced. And then in the end, we get a standard deviation, so meaning a dynamic, whatever changes there is really very, very small for PIP2 bound at the same time. Both sides have to be occupied, and then you get very much boosted effects. We also performed coarse-grained simulation, which is pulling the channel from, or the voltage sensor from one state to the other and measuring the force required to pull. And when we did this in both directions, so in the deactivation and the activation direction, you can see that here in blue versus the orange side, you can see when both PIP2s are there, you need less energy. So it's really removing like an energetic barrier between two open states. Okay. So now I take a little bit more time. So there's something else. There's a benzodiazepine which activates the channel as well at 1 micromolar. So in another study I'm working on since many, many years. So what we first did is we wanted to identify the binding site. So we scanned the pore domain, and we identified some residues which really had an impact of the inhibition. Here in green are those residues which have an impact on the inhibition. In red is the compound docked to the pore domain, and you can see there's a pretty good overlap there for binding and what we have identified by the alanine scan. So, but that was only the part of the study which we published a long time ago, and it just accommodated the results from here in magenta S5 and S6. And modeling could only be done because there was only KCSA structure at that moment. There was not even a voltage sensor included in the structure provided by McKinnon. But then later on, the outer part, this benzodiazepine part, this is an indole sticking in between there, and this benzodiazepine part is interacting with a 310 helix, the lower part of the S4. So we also checked that by mutagenesis scan, and we identified that really there's an interaction with the lysine there. And then we also, in this model, when we had this model docked there, we also identified that there might be an interaction with the S4S5 linker. We mutated also this interacting part, which is a tryptophan. Exchanging by an aromatic reduces the function, either neutralizing by an alanine mutation or introducing even a charged one reduces even more the activating effect of the compound, confirming the model. Okay, so here you can see the compound sitting in there, and it's really like forming a glue between the lower voltage sensor, the pore domain in the back, and also the S4S5 linker, so really crossing the whole thing. The compound is able to also activate LQT-associated channels, so activation, partial recovery can be obtained here, indicated by the white color. However, there's one mutation which is resistant. How does that work? Well, there's a repositioning by the mutation, and an aromatic ring is repositioned. However, when the aromatic ring is repositioned due to the steric hindrance, then we get steric hindrance with the indole of the compound, so it's virtually pushed out of the binding site. But we can also understand why a compound in a genotype is no more working. Then we wanted to figure out what this compound is doing. So we did mathematical modeling, and we could give it a forward rate, which is increased by the compound stabilizing the channel. But we also did some current recordings, and we wanted to see whether the voltage sensor is stuck. I'm close to finishing. So what we did is we used voltage clamp fluorometry, and what you can find is that there is really some immobilization of charge at the voltage sensor having there. So the compound is really doing something similar to the PIP2. So I would like to summarize. So we have identified a novel PIP2 binding site for another PIP2, which might be involved in trafficking of the channel. We have shown that a compound which uses a similar mechanism can be used to activate LQT-associated channels. We can explain why a channel is no more sensitive to the compound, and we could identify the binding site and mechanism of this compound. So we had some collaborators, and I thank you for your attention.
Hi there. Thank you for your talk. Could you elaborate a little bit more on the physiological role of that endosomal pool of channels that you identified? Is it relevant just to cardiac cells or all cell types?
I would expect that it's in all different kind of cells, and it's a trafficking effect. So this channel is then incorporated or increased in function as an effector of chronic stress, basically. So SGK, the serum and glucocorticoid kinase, couples it to chronic stress. So this channel is an effector of acute stress via beta-adrenergic signaling, and also chronic stress, which is pretty interesting, I think.
Do you have a feel for what sort of percentage upregulation of the current this will actually result in?
So in several systems, it was something like 200 to 300%. So it's massive.
Wow.
And it's really enriched in these vesicles and sitting there, waiting there for it to be transferred and incorporated into the membrane. It goes like this. And SGK is also an immediate early gene, so it's really a fast response.
Thanks.
Well, many thanks for the exciting talk. So, as you say, this is an endolysosomal PIP2, which has got its own channel and other things. But is there any known LQT mutations where this particular site is mutated so that the problem is in the trafficking rather than functional loss of function?
Yeah, I showed them. There are 3 of them which really don't respond to the lipid anymore or are even inhibited by this kind of PIP2. Yes, there is. And interestingly, also other targets which are included in these vesicles are also hold back. So you will mess the ion channel composition at the plasma membrane when you have mutation in there. But you also have an effect, for example, on HERC and other channels. So it's becoming very complex then.
And on those mutant channels, do you see them visually trapped in the endolysosomal membrane a lot? Or?
Yes.
Okay, great, thank you.
Hello. Lovely talk. That cysteine scanning study you did on the S0 helix, would you be able to get more information maybe by doing some MTS-style reactivity against some of those substituted cysteines?
Yeah, we were starting actually with charged MTS to cross it to the, or glue it to the membrane. Yeah. But we killed our oocytes with that. So that's why we ended up with cadmium, and there we got something like 50% of oocytes who got leaky. So these experiments were a pain in the butt.
Sure. Well, thank you.
Okay, just a basic question because I don't know the channel. About the position of the S0, yes. Right? Are there hydrophobic amino acids like some S4, S5 linkers that keeps the helix like packed against the membrane? And in that case, so PIP1 would further help positioning the S0, or if it wasn't for PIP2, this helix would be like free, you know what I mean? Like.
Yeah, so based on the modeling, you really can expect that it stays at the plasma membrane. In the center, there's tryptophan, and tryptophan are classically enriched in the inner leaflet of the membrane. So, and it's basically more or less in the middle, which really makes it like Wippe in English, you know, like where the children sit on, something like that. Yeah, something like that. So there seems to be something really about the mobility relative to the plasma membrane. And then there are a few chargers like arginines, which also might have an electrostatic interacting impact with a membrane. Because it's really just at the, it's really like.
Yeah, Gizi, nice talk. Are there sites for PIP3-5 on KCNE1? Because when there are PIP4-5 sites on KCNE1 as well as on KCNQ1, and then when it assembles, when it reaches the membrane, does KCNQ1 still endowed with the PIP molecules that were necessary for the trafficking?
Yeah, so there's one mutation indeed in KCNE1, an LQT-associated mutation which disrupts the PIP2 sensitivity for this PIP2 as well. So which really indicates that there's either a direct or indirect effect. But due to the fact that we are not really sure where we put in the model the transmembrane helix of KCNE1, it might be flubbery and swinging around there during gating, we couldn't really model it. So I thought it's not precise, so we left that out. Just for that reason, just for technical reasons. We knew we would not be able to pinpoint it really down. Yeah, and this D85, you know that. It's a D85 mutation, which is disrupting, which is also at the intracellular border there. But what it really does, I mean, we all guys can talk about that, and you might have some results from 25 years ago in mind. I forgot about it. So.
It's time for us to turn to the next talk.
Thank you.
Very thankful.